All right. Welcome everybody to our August MicroTalks. So just a quick reminder that we have this seminar series at the last Wednesday of every month at 4 p.m. The topic of this session today is all about phage, a friend or foe. We still have a few slots left for if anybody wants to sign up to give a talk sometime this academic year. I'll post the link in the chat if you want to do that. And we are also having a gift card raffle for us, all the attendees at this talk. So if you want to enter the Starbucks gift card raffle, you can enter your email in the chat. All right, so let's get started. First up, we have Jacob Zulk, who is currently a Ph.D. candidate in immunobiology and microbiology at Baylor College of Medicine, and he's working in the Maresso and Patras labs. Today, he's going to tell us about the fitness costs associated with bacteriophage resistance in uropathogenic E. coli. Take it away, Jacob.
All right. So thank you for the introduction. As was said, today I'm going to be talking about kind of uropathogenic E. coli and how we can use bacteriophage in the treatment of UPEC infections. All right, so partially the reason we study UTIs is because they are such common bacterial infections. So worldwide, there's about 150 million UTIs each year. And so overall, what this means is that about 1 in 2 women are going to have a UTI during their lifetime. And while that's a huge number, there's also a subset of those women that'll go on to have recurrent UTIs. So these are more UTIs with the same pathogen, just essentially recurring after initial treatment. So overall, the majority of these UTIs here are caused by uropathogenic E. coli, so somewhere on the level of 70%. So this makes an appealing target when we're looking for new therapeutics, because we can target really one type of bacteria for that'll help a lot of these infections. So I don't think I probably need to go into huge depth here with this crowd, but antibiotic resistance is a growing threat among all bacteria, but especially or including E. coli. So there's an estimated almost 3 million infections with antibiotic resistant bacteria each year. And a portion of these are caused by what we would consider a reservoir of E. coli. So these are the Enterobacteriaceae. So these are things like Salmonella, but also E. coli, which includes uropathogenic E. coli in there. So as you can see, there's a large burden of disease, and there's an increasing antibiotic resistant threat amongst all E. coli, but urinary tract infection causing E. coli as well. So the question is really, if antibiotics aren't doing the trick anymore, and if they have a potential to not work as well, do we have anything else? And as I think everyone can guess, based on the topic of today, we think that bacteriophage or phages could be an alternative to antibiotics for treating some of these infections. So some of just the benefits that phage have when we're comparing them to standard antibiotics. So standard antibiotics take somewhere in the billions of dollars to develop, and then antibiotic resistance can quickly develop. So because of that, one of the real benefits of bacteriophage is that they're a lot quicker and cheaper to develop. So this is HP3 here, which Sabrina, the next speaker, played a hand in isolating. So HP actually stands for Herman Park. So this is actually isolated from goose feces in a park, which is pretty cheap when you compare it to standard antibiotics. So it's a lot quicker to develop one of these and isolate one of these than a standard antibiotic. And importantly, when we're thinking long-term, bacteriophages are relatively specific for certain bacteria. So rather than kind of the antibiotics where you might target a whole gram-negative group of bacteria, bacteriophages are a little bit more specific. And so maybe we can decrease some of the off-target microbiome damage that we're doing when we're treating infections. So we really wanted to know, kind of as our first question here, can we use these bacteriophage to actually kill uropathogenic E. coli in urine? So a lot of people have looked conventionally at the ability for phage to work in things like LB media, where it's really nutrient rich and these bacteria are growing well. There's very little for the phage to get stuck on. But we wanted to know, can we use bacteriophages in the context of urine? So to do this, we selected 2 UTI-causing UPEC strains. So UTI-89 is a little bit more of a conventionally used uropathogenic E. coli strain that was isolated in the mid-90s, I believe. And then DS566 here is a more conventional isolate. So this was isolated from the VA Medical Center here in Houston just about 2 years ago. And so we tried one of our phages in our lab, ES17, against both of these strains, looking at the bacterial growth of 96 well plates containing either bacteriophage ES17 or no phage. And so we did this overnight cultures in a plate reader. And what we saw was that for the first 6 or so hours, there's really this suppression of any growth in these wells that contain the bacteriophage. And so compared to the no phage control wells, which go really well in urine, we saw essentially no growth. But at some point, there's this resurgence of growth in these even phage treated wells. And so what happens is if you take these bacteria that are growing in these wells that have been treated with bacteriophage at 18 hours, and then retry them again and challenge them with phage again, what you get is growth that's much more similar to these no phage control wells that you initially see. So we wanted to know what's really causing this change. So what's taking these bacteria from a population that are susceptible to bacteriophage to ones that are no longer able to be infected by these bacteriophage? So what are the actual mechanisms that they're using? And importantly, what are the consequences of that for both the person and the bacteria themselves? So for this first question, in order to actually ask, how are they evading ES17? What we had to do is isolate these bacteria that were resistant. So to do this, we did kind of a modified system of what I just showed, those growth curves. But instead of measuring optical density, what we did is we challenged the bacteria with bacteriophage ES17 overnight. And then we plated these lysates, or the bacteria that were still present in the liquid after the overnight culture, onto agar containing ES17, phage ES17. We then went the following day, picked colonies from these and put them back into the same system, right? So we're essentially serially passaging these bacteria against the bacteriophage. And so we're generating a clonal, highly resistant strain. And so after we run this back through, when we get to this portion where we're plating on phage top agar, we take these out and send them for whole genome sequencing to look for what changes actually occur. So what we see when we do that is that all of these bacteria have mutations in lipopolysaccharide, or LPS. So wild type LPS has a few different components. There's this lipid A portion, which kind of anchors the LPS actually in the outer membrane. And there's an inner and an outer core followed by an O antigen, which is the part that's a little bit more variable between the pathogens. And what we saw when we isolated 4 different strains that were resistant against ES17, so 2 UTI-89 strains and 2 DS566 strains, is that they made truncations in LPS on the genome scale. So I will say we currently haven't actually looked at the production of LPS and see if it's different, although it does just by phenotype look like there's differences and changes in these. But what we saw was these 2 UTI-89 strains that were resistant against ES17 both had mutations in RFAH, which is a transcription factor important in actually assembling LPS. And then DS566, there were 2 mutations, one in the outer core and one in the inner core. So this DS566-2 is what we're calling it. It's kind of the one that has the largest truncation actually present in it. So we wanted to ask kind of what are the actual functional consequences of this? So we looked at what we actually know LPS is important in. So when we looked at things and kind of in the literature, there was quite a few things that stood out about what was known about the importance of LPS. So specifically, people had looked at, for sure, the growth and colonization and the ability for these bacteria to actually stick around when they have truncated LPS. And also people have looked at the immune response as well as antibiotic resistance and kind of how, when those are affected, antibiotic resistance is affected. So what we aim to do is kind of start from the basics. So how do these grow? And so what we did is we just did overnight growth curves again, looking at our two mutants and the wild type for each bacterial strain. In the presence of urine, these bacteria grew more or less what the wild type did. You'll see in this DS566, which kind of stands out, is that there's kind of a slight growth effect in these two mutants compared to the wild type. But what you do see is still you see the lag log and then the stationary phase here. But interestingly, this is in LB. When you look in urine, you actually see a pretty severe attenuation in all four mutants. So while they're able to grow more or less the same as the wild type in LB, in urine, there's kind of this severe defect in their actual ability to grow and to actually continue to grow. So this is good news for us if we think about in the context of the actual urinary tract infection, where if we were to drive these changes, perhaps these bacteria would be unable to grow as well as they actually would be normally. And so, at least from myself and most people I know, we don't have 96 well plates as bladders for the reservoir of these bacteria. So we wanted to look at a little bit more inaccurate kind of what we would actually see in vivo. So to do this, we used a pretty well-known mouse model of UTI. So in this mouse model, you essentially take a needle covered with catheter tubing, and you essentially make a catheter in order to inject the bacteria directly into the bladder. So you go straight up through the urethra into the bladder, and so your bacteria are actually getting in there. And so for our setup, we did that with our wild type and then one of the two mutants for each strain. And then at 24 hours, we collected the bladder, kidneys, and urine to assess colonization. So I'm going to show bladder here just because it's most easiest to kind of understand with the least variation. And so in the wild type strain, you can see UTI-18 is a UTI former. And DS566 is less so, but you still have bacteria present at all those times. But when you look at the mutants of these strains, what you see is kind of a pretty profound colonization defect. In the UTI-89 mutant, this is one of the ones with the truncation and kind of the outer portion of the inner core. You see about a 2 log decrease in the colonization of the bladder at 24 hours. And in this DS566 mutant, what you actually see is a complete absence of any bacteria in the bladder at just 24 hours. So again, this is good news. Not only did we show earlier that these bacteria that are resistant to ES17 are less able to grow in urine, but they're also worse at colonizing, it would appear. And so looking forward, it's probably unlikely that we're immediately going to make the change straight to phage from antibiotics. And what's probably more likely, especially since some of these bacteria that were resistant were still able to colonize, is that we're going to have kind of a combination of antibiotics as well as phage used. And so we wanted to see specifically what effect having phage along with antibiotics, so testing our phage resistant strains for their antibiotic susceptibility. So to do this, we did MIC assays, minimum inhibitory concentration assays. And so this will tell us at what concentration of antibiotic the bacteria are still able to grow. So we compared the wild type and the two mutants for each strain in their sensitivity to the antibiotics colistin and polymyxin B. And our rationale for choosing these two is they're both antibiotics that work on the bacterial membrane itself. So the thought being that if we're making mutations to LPS, which is all over the surface, perhaps we're opening the ability for these bacteria or these antibiotics to actually get to the bacterial surface. And what we see when we look at that is we do see a decrease. So In these UTI-89 strains, we see about a two-fold difference in the MIC between colistin and or the wild type and the two mutants. And then when we look in the DS566, we kind of see it's kind of reminiscent of the mutations themselves. You'll remember DS566 had just one mutation that was in the outer membrane. And then DS566-2 had the mutation that was pretty drastic right in the inner membrane. And so it seems to be that the larger the mutation, the more susceptible they are to antibiotics. And this pattern somewhat holds true with polymyxin B as well. Again, it's a pretty similar to the previous one, where it looks like probably about a two-fold decrease for the original wild type, and then the largest susceptibility is in this population that has the largest LPS mutation in it. And so in conclusion, kind of what we've seen is bacteria might become resistant to the phage, but a way what we can get around this resistance is to look for mutations that come at the cost of bacterial fitness. So mutations that the bacteria have to undergo that essentially renders them either less effective in their environment, in the urinary tract, for example, or leaves them susceptible to things like antibiotics that we're going to use in combination with this treatment. And so one thing that I will say is an obvious caveat for this is that I showed you kind of the phenotypes or the phenotypes of these bacteria that were isolated in vitro and then used for all this in vivo work. I think the first question that we need to have is how effective can we actually be at driving these mutations in vivo? So if we're obviously not driving these mutations in vivo, that kind of rules out this line. But what we see is, or kind of how we're going about addressing this, is essentially a phage therapy treatment of mice. So what we do is we take our mice, we give them UTIs the same way as we did previously, and then 4 hours later, we follow up the same administration protocol by giving bacteriophage. And then the following day, we look at the bladder, kidney, and urine burdens again. And so to do this, we used bacteria UTI-89 that I discussed earlier, and ES17. And what we see is when we treat UTIs with ES17, we get near sterile urine at 24 hours in the majority of these mice. But there's a subpopulation of these mice that still have pretty high bacterial burdens in the urine at 24 hours. And so what you wanted to know is, are these actually resistant? Are we actually driving these mutations in vivo? So we looked at one of these mice specifically, and what we did is we picked 10 colonies that we got out of the urine at random. We put these on agar plates and let them grow. And then we spotted ES17 on top. So if there's clearance like there is here, that would suggest that these bacteria are still susceptible to the phage. And if there's not, that would suggest that these bacteria are actually resistant to the phage. And so what we saw in that one mouse is that 40% were actually resistant to ES17 by that measure. So that suggests that we are driving some of these mutations. However, there's still work to do if we actually want to get to that 100%. And I think there's kind of a whole litany of different ways you could address this. Mice obviously do not really listen when you ask them not to wake up from being anesthetized and immediately urinate out the phage you just gave them. So perhaps this is more of a model kind of model, part of the model that's just never going to be quite perfect for us. We're also only giving one dose of bacteriophage, so perhaps it's just a multiple dose thing. And what we also didn't check, we're looking at 24 hours here. So I think that another kind of thing we could look at is just, is there a quicker time to resolution in these bacteriophage treated mice to look to see if we're actually driving kind of a self-limiting infection comparatively. Other further directions, we're looking at how we can combine antibiotics and phage in vivo. I showed that in vitro data where they were already resistant. What we're looking at is, can we use maybe a lower dose of antibiotics or a shorter duration of antibiotics as well as phage to increase the actual clearance of bacteria. And finally, we showed that LPS mutations left the bacteria sensitive to both the urinary tract environment and to antibiotics. But the other question is, are there other targets that we can look for? It's certainly not the only thing expressed in the outside of UPEC is LPS. So can we actually look for other bacterial, adhesions that we can target with bacteriophage in order to make the bacteria have to choose whether they, say, colonize well or are susceptible to our bacteriophage cocktail. And with that, I just want to thank everyone in the 2 labs that I'm in. Justin here did a lot of the actually assembly work to find out what these mutations were. And I just want to thank everyone who helped with both the dissections as well as the procurement of the miscellaneous phage that we're using and the urinary pool that we are using. And with that, I can take any questions.
If anybody has any questions, you can type them in the chat or ask directly.
Heidi?
Yeah.
Oh, go ahead. Somebody else.
Yeah, I had a question, Jacob. How do you envision it in delivering the phage, say, to a person?
Yeah, I think that's a really good question because the majority of people are not going to want to be catheterized to get phage. I think currently what we're trying is really just seeing how many phage we actually need to get into the bladder. Obviously, the easiest way would be a delivery method that's just orally or something. Currently, there's a clinical trial going on in New York, I want to say, that's looking at different ways to give bacteria or bacteriophage. I mean, one of the ways is an IV injection, which I would say is not that much less invasive than this quick catheterization, especially for one of the large populations that I think this could be useful for is these recurrent UTI patients. And I think that those people would probably be fine with a one-time catheterization if you had something that worked well. But currently, I mean, we're just really looking at if we give this much, will we actually treat the UTI?
Yeah, absolutely. That makes sense. Yeah, thank you.
I think you've got a question in the chat.
Yes,
And then-
I see it.
Oh, yeah.
I have a question. Yeah.
Okay.
Why don't you do that?
Yep. So the question I'm seeing is phenotypic differences in the truncated LPS strains. Yes. So there are a few. I will say the one that's probably the most pronounced that I've seen a few times now is that if you grow these bacteria stationary, what tends to happen is a lot of them kind of crash out, or not crash out, but they don't stay in suspension as well, which kind of makes sense if you think about you're removing portions of LPS. So you're essentially changing the charge surface of these bacteria or kind of what's exposed to the aqueous solution. So I think that might be part of it. We're looking at if biofilm properties change as well in these mutants. So far, it's mostly that there's no huge differences, although that's something that obviously needs to be seen before you would put this in a patient. You don't want to drive the biofilm.
Jacob, I have a question about the resistance that you saw. I was thinking that if you had 2 phages instead of just one, and that they had different mechanisms, would you think that that would help you to not have the resistance come up?
Yeah, I definitely think so. I think the 2 that I looked at coincidentally ended up both that I tried to initially put together, both ended up being against LPS. So that was my original thought, but both of them ended up being LPS strain or targeting. So, but yeah, I think there's plenty of things that are important for adhesion to the bladder wall, say, and I think if you could target that, you could perhaps prevent a lot of the actual infection as well.
The other question I had was, in your mouse experiment, you had 40% of the the E. coli that grew were sensitive or resistant to your phage. So 60% were sensitive. What do you think the other was the deal with the other 60%?
Yeah, so I think, well, one option I can think of easily is that UPEC can actually invade the bladder epithelium. So it's perhaps that these bacteria were able to get into the bladder in that 4-hour period that we gave them before we gave phage, and they're just essentially unreachable by the bacteriophage themselves. Another thing I think, I mean, the mice themselves, as anyone who works with mice can probably tell, do not, I mean, once you give so much liquid into the bladder of these mice, they're inevitably going to wake up and urinate. So there's probably some level of just they're getting rid of it pretty quick. There might just not be enough time for it to, the two to reach each other.
Great. Really nice talk. Wonderful work. Very exciting.
Thanks.
I think you have a couple more questions in the chat, and I believe we have time. So first from Rodolfo Contreras, he asked if the mutated strains are grown in urine media several generations, do you think the fitness loss could be reverted, and would the strain still be resistant to phage?
Yeah, I think there's a chance they could. They kind of had different mutations. So there were a few that were premature stop codons. So theoretically could revert, probably a little less likely. There were a few that were, I don't remember the exact mutations. There is, I think if they're not being challenged with phage though, they're probably unlikely to have any really driving factor to keep them one way or another. So maybe, I guess is the best answer to that. Something that would definitely need to be looked at though, if you want to actually translate this into a person.
And Mohammed asks, is there any baseline resistance to the phage in UPEC if it's not pre-treated with phage?
So far, no, that we've seen. I mean, there's some strains that lack, say, O antigen, that maybe would be a little bit resistant if that's where we're thinking it might interact. But so far, no. If you just take the wild type E. coli that we're growing and treat them with phage, they're pretty much susceptible, all of them.
Great, thank you so much, Jacob, for sharing your work with us.
All right, thank you.
So next up, I'm happy to introduce Sabrina Green. Sabrina graduated with her Ph.D. in molecular virology and microbiology at Baylor College of Medicine, working in the Maresso lab. She then co-founded the TAILFRing Service Center, and she is currently a director of research and development there. So today she's going to tell us about tailoring phage for the gut and beyond.
Okay, can you hear me and you can see my slides? Okay, great. So today I'm going to talk to you about a little bit about my research and how we're actually using this knowledge to help patients right now through our service center, TAILFR, at Baylor College of Medicine. So first, I'm going to introduce you to a problem I'm sure many of you are aware of, and that is the invisible pandemic, otherwise known as antibiotic resistance. And this term was not coined by me. It was actually coined by the World Health Organization. They also call this a global epidemic. The CDC refers this to an epidemic as well. And so this is a worldwide threat, but here in the US, it's particularly bad. 150 to 160,000 people die per year due to multi-drug resistant bacterial infections. And it's said to get worse with the current pandemic that we're on. So I study a particular type of pathogen. It's an E. coli, as Jacob introduced you to UPEC, which is a subset of X-Pac, so extraintestinal pathogenic E. coli. It's the #1 cause of adult bacteremia, gram-negative. It's the #1 cause of urinary tract infection, #2 cause of neonatal meningitis. These severe infections can lead to 80,000 deaths per year. And these bacteria actually live inside of us. So the #1 reservoir of X-Pac is the human gastrointestinal tract. And they don't cause intestinal disease, they only cause extraintestinal disease. And there's a clonal group of X-Pac, it's called ST131, and they've actually been linked to the global spread of antimicrobial resistance. So these strains are colonizing us and potentially spreading antibiotic resistance to other bacteria. And there's no vaccine against X-Pac. So how do we actually treat and prevent these infections? Well, for that, we turn to phages or bacteriophages. Some have referred to them as a perfect predator. They are viruses of bacteria. They've been shown to be effective in clinical cases. One good example of this is this case, Dr. Tom Patterson. He developed a bacteremia due to this bacteria, Acinetobacter baumannii. And if it wasn't for these phages that he received via IV treatment, he would not have survived the infection. And he's alive today thanks to this phage therapy. So we know a lot about phages. There's been a lot of research. They are discovered in the 1900s. They're still used in Eastern Europe. They're used on a compassionate use basis here and in other countries. So there's a lot of research about them. We know, for instance, they're highly specific. So they bind to receptors on the bacterial surface. They can kill multi-drug resistant bacteria, injecting their DNA, creating more of themselves, and lysing out the bacteria to then infect other bacteria. They've been shown to be safe in clinical cases. And more importantly, they're evolvable. So they can evolve to overcome resistance. But we also know a lot of disadvantages to their use. They can be highly specific, so certain infections may require multiple phages. You may develop an immune response to phage depending on how it's administered and for how long, and depending on the phage sometimes. And then bacteria become resistant, but again, we may be able to counter this resistance because of the evolvability of the phage. But there are some unknowns. So we don't know what exactly is happening at these body sites. So we know that the phages can kill bacteria in culture, for instance, but are they killing bacteria in the blood? Are they maintaining specificity in the gut? Do we see resistance at these sites? These are really important questions that we should answer before we try to implement phage therapy for these resistant bacteria. And so the main focus of my research was actually in the gut. And so what we did is we utilized model systems that we developed in our lab, a mouse model system, ex vivo systems, human intestinal enteroids, to test to see if we can use phage to specifically target these bacterial pathogens to remove them and prevent antibiotic resistant infections. And so the point of this work is to understand how bacteria and phage interact at these treatment sites to develop better therapies and preventatives. And so our first task was to actually find these phages. So originally we did not have access to sewage, now we do. And so we kind of worked with the low-hanging fruit. We worked with what samples we could get, and that was bird feces and dog feces, which are everywhere. And these E. coli actually have shown to colonize birds and dogs, and they're actually reservoirs of these E. coli as well. So we use these samples to isolate phages. So this is an EOP chart, and this column on the left side are our ST131 clinical isolates, and the top row are all of our phages. And so the black squares indicate that the phage could kill really well, and the gray squares indicate that the phage could kill moderately well, and the white indicates that the phage could not kill. And you can see from this chart that we were able to isolate several phages that could kill these bacteria in these plate culture systems. And you can see that we had a variety of phages. So we found the phages. Now, do they actually work in these model systems? And so we tested this in a bacteremia model that we developed in the lab. So in this model system, we take a mouse and we inject them IP intraperitoneal with a high dose of our E. coli. And an hour later, we inject them IP with a high dose of one of our purified phages, HP3 here. And after an overnight infection, we look at the health of the mice, and then we euthanize the mice and we plate organs for this E. coli. So this is a disease severity chart where 0 indicates that the mice were not sick, and a 4 indicates that the mice were moribund or very sick. And you can see in the mice that did not receive phage, they became very sick. And those that did actually did very well. And when you look at the organs of the mice, and this is in log CFU per gram of tissue, you see about a 3 to 4 log reduction in bacterial burden at these major organs. So we can use phage therapy to reduce disease severity and bacterial burden and multi-drug resistant bacteria. But what about the gut? So the gut is an incredibly complex place. You have the entire microbiota, you have the mucus where bacteria could be hiding, epithelial cells, immune cells. So you can imagine it may be a hard place for a phage to find its host. But we tried it anyway. We used a mouse colonization model this time. So in this model system, we take mice and we give them an oral dose of our E. coli. We can colonize the mice for several weeks. And after a day of colonization, we then start to give the mice the same phage, HP3 here, that worked in the bacteremia model every day at a 10 to the 9th PFU dose. We also had a group that received the phage in the water, and we had a group that received an antibiotic in the water. So this particular E. coli was susceptible to an antibiotic, so we used that as a control. So looking at colonization over time, this is bacterial levels and log CFU per gram of fecal pellets. You can see that even though we gave phage either gavage or in the water, we saw no significant difference between the groups that received phage and those that did not. But we did see a precipitous drop of E. coli when we gave the mice an antibiotic. So in this case, the antibiotic did work. And no detectable CFUs throughout. At the end of this experiment, when we euthanize mice and we plate the small intestine and the large intestine for phage, we found in those that received phage either via gavage or in water, there's still phage present at these sites. But when you look at the bacteria, the E. coli present at these sites, we see no significant difference between the groups. Only in the group that received the antibiotic, we see no detectable CFUs. So this told us that the GI may actually be a prohibitive environment for phage infection, and we wanted to figure out why that was true. So we took this out of the mouse into a more manipulatable system. So this is an ex vivo model. In this model system, we take mice that are healthy and we euthanize them, we remove the cecum, and we take the contents and we homogenize it in saline and use that as growth media, nothing else added. We add our bacteria and phage at the same time at an MOI of 10, so 10 times more phage than bacteria. And then we selectively plate for our bacteria and we plaque for our phage. So looking at one of these growth assays, so we did this in LB, and you can see that our E. coli grows really well in LB, and when we add phage, it kills. When we take fecal pellets from healthy mice and homogenize that and try to use that as growth media, we see the same trend where the phage killed really well. But when we use cecal media, we see no significant difference between the groups. And when we look at phage levels with this media, we see there's significantly less phage in our cecal media. So the phage are not growing and they're not killing their host. So this told us that there are factors that are present in the large intestine that are inhibiting phage. So we were able to take this cecal media and filter it with a 0.22 micron filter, and the phage started to kill better. We were able to boil the cecal media and use that, and the phage started to kill better. And we used a cocktail of antibiotics to kill off the rest of the microbiota, and the phage did not kill really well. So this told us that this inhibitor is likely a large protein in your large intestine. So we turned to the largest proteins in your large intestine, the mucins that make up your mucus layer, specifically the gel-forming mucins. So I'm not really good at drawing And I didn't like any of the drawings of mucin because they were very complex. So I like to show this bottle brush as this representation of a mucin molecule. And actually the gel forming mucins are more like two bottle brushes put together head to head, where the bristles are the glycosylated regions, the sugars, and the handles are the non-glycosylated regions. And it's within these handles that these mucin monomers come together, forming disulfide bonds to create these large polymers that make up your mucus layer. Bacteria have been shown to bind to these mucins, and sometimes that's how they colonize. And phage too have been shown to bind to mucins using capsid proteins. But what's not known is whether these sugars or these glycoproteins are inhibiting phage infection in the gut. So we wanted to use our system to figure out if this was true. So what we did is we took our cecal media and we centrifuged it at a high speed to pellet these large glycoproteins. And we resuspended that and call that our insoluble cecal media. We also used our supernatant in our growth assay. So again, this is an LB. You can see that the phage really kill well here in the red. In our cecal media, no significant difference. So you see the inhibition. In our insoluble media, you see some killing, but some resistance. But then in our soluble media, where the glycoproteins, these large glycoproteins are either not present or present in low amounts, you see suddenly the killing is restored. And when you add N-acetylcysteine, which cleaves these disulfide bonds in these mucins, you see suddenly that the killing is restored in the insoluble media. And when you add purified mucin to the soluble media, you see this inhibition again. So this told us that mucin is inhibiting phage infection. So we know that there is a large variety of phages in the world, and maybe this is not true across the board for every phage, and maybe there are phages that have adapted to actually utilize these sugars. And so knowing that, we screened our phage library for a phage that was capable of killing under these conditions. So this is a growth assay in LB plus 1.5% mucin. And these are all of our phages that could kill our bacteria. And we could only find one phage, which Jacob talked about, ES17, that was capable of killing under these conditions. And when you look at this same library growth assay in LB only, you can see that there were several phages that were effective at killing this bacteria in LB, but not effective when mucin was present. And ES17 really wasn't the most effective in LB only. So we thought this was really interesting. So we took this phage and we tried to figure out what was going on. So we performed an absorption assay. So absorption is when phages, they attach to the bacterial surface and inject their DNA. And we can measure absorption by incubating phage and bacteria together and taking samples and centrifuging them and plating phage. And so this is free phage, which should reduce over time. But we modified this a bit. We took our E. coli and we incubated with mucin. Then we centrifuged and we washed. And so essentially coating our E. coli in this mucin matrix, and we perform this absorption assay. So this is ES17. I'm not sure if Jacob showed you this image, but it is an unusual looking phage. Yeah, it has this elongated capsid, kind of like a corn dog, I've been told. And these short tail fibers, it's a podovirus. And the tail fibers are actually paddle-like too, so it's a bit unusual looking. And this is the absorption assay of ES17. And you can see that when 0% mucin is present, 0% of the phages are absorbed in 10 minutes. But when we add 1.5% mucin, you see that 98% of the phages are absorbed in 10 minutes. And when we perform the same assay with HP3, the phage that was not effective in our mouse model, you can see that it goes from 98% when no mucin is present to reduce to 84% when mucin is present. We've also found that this phage was very effective in ELISA assays that with mucin present. So we thought that our phage was actually binding mucin significantly better than HP3. And so this led us to believe that this phage may be utilizing mucin as a receptor. So this is an unusual thought because mucins are mammalian sugars. And we thought this might be true because of something called capsule. Capsule does something, or bacteria do something called molecular mimicry, where the bacterial capsule can mimic our human sugars. That's how they colonize us, and they evade the immune system and can continue to colonize us. And so phages, of course, have adapted to utilize capsule as a receptor. They can bind to it, degrade it. Many phages that are effective in biofilms have this special ability. So going back to this idea of molecular mimicry, E. coli have this capsule type. It's called K5, otherwise known as heparosin. And so they call it heparosin because it's very similar to heparin sulfate. So heparin sulfates are sugars that are present on almost all of our cells, our epithelial cells at the mucosal surface, heparin sulfate proteoglycans. And so the tail fiber of ES17 here in the blue actually is very similar to the tail spike in a K5 phage, particularly in the area where it's binding the receptor, so the bacterial receptor. Here in the yellow, those are areas of homology. And so what we thought is that this phage, K5, which you can imagine what it's binding to, its receptor is a K5 capsule, is very similar to our tail fiber in our phage. So we think that our phage has the ability to bind K5, and because it's so similar to heparin sulfate, it also has the ability to bind to these heparin sulfate sugars. So this is just a hypothesis, right? Well, is it actually true? So what we did is we cloned this tail fiber, we purified it, and we performed a microarray assay at Emory University. And so this is a microarray chip where there are glycans present, 860 different glycans. And what they do is they add your protein and they use antibodies to detect whether your protein is binding the specific glycan. And so the output is relative fluorescent units. And you can see in this top graph that a lot of our data was nothing, no signal. And really the only signal that we got here in the red bars and the graph below that were for glycans that were specific for heparin sulfate. So we think that this ES17 tail fiber is binding heparin sulfate, but is it binding it in the gut? So for that, we use these human intestinal introids. These are derived from crypts and human patients. We can form them into 3D organoids and then form them into monolayers so that we can easily infect. And they're multicellular, so they actually do make mucin, and they make these everything that you have in your gut. And so what we did is we added our phages to these monolayers, and we washed and then we stained them. And so we have antibodies that were directed towards these phages too. So this is a monolayer of our colonic introid. And you can see here the blue are the cells and the red is MUC2. This is a dominant mucin in your colon. And when we add phage here, so the phage is in green, you can see that ES17 just covers the entire monolayer, even where this MUC2 is present. And when you look at HP3, really we see no signal. So the HP3 doesn't seem to be binding. So ES17 binds these human intestinal introids, but is it dependent on heparin sulfate? Well, for that, we use an enzyme, heparinase sulfate 3, that cleaves this heparin sulfate at the area where we think that ES17 is binding. And then we added our phage. So you can see on the left side, again, untreated, this phage completely covers these introids. But then when we add the enzyme and then we add the phage, you can see there's not much green. So the ES17 is not binding. And you can graph this and you can see that there's a significantly lower ES17 present when this enzyme is added to these enteroids. We've also found that this phage, even though they're bound to the enteroids, can still kill E. coli. So it still has the ability to infect and kill E. coli. But going back to our original question, can we use this phage to eradicate these bacteria in the gut? So this is our original failed experiment, right, where we colonize the mouse with E. coli, and the next day we give them a dose of our phage. This time we use HP3 and our new ES17 phage. And we also gave them 10 times more phage than we previously did. So the idea is, like, could we just overcome this barrier by adding more phage? So this is bacterial levels in the small intestine and the large intestine. And you can see that with HP3, there's no detectable CFUs in the small intestine of the mice. So adding 10 times more phage seemed to help in the small intestine. And when you look at ES17, we still had one mouse that was colonized. Now, when you look at the large intestine, you can see that there are still more mice that were colonized in this HP3 group. But in the ES17 group, we only saw one mouse that was colonized. So it seemed like ES17 was just as effective in the large intestine as it was in the small intestine. And we think it's because of its ability to utilize these mucins that are present, more present in your large intestine to better infect these bacteria. So in summary of some of this research, we utilize these great model systems to see if we can effectively eradicate these E. coli from the gut of the mice to prevent infections. And with these model systems, we learned that there are some barriers to phage therapy in the gut. The major barrier being the mucins, or the sugars that make up your mucus layer. But knowing this, we can actually screen for phages that are effective at utilizing these sugars to better infect the bacteria in their gut. So we could potentially use phage to treat these drug-resistant infections and to remove this reservoir to prevent infection and the spread of resistance. So what are we doing to help patients now? So we started this program. It's called TAILFR. It's Tailored Antimicrobials and Innovative Laboratories for Phage Research. We are made up of researchers, clinicians, and technicians. And what we do is we work with clinicians at the Texas Medical Center and other centers, including the Mayo Clinic and UC San Diego. And these are patients that they're on their last drug. They have they're antibiotic resistant infections. They have no other option. And so we develop phage cocktails for them. So typically a clinician will contact us. We go through our library, we find phages that are effective against this bacteria, we characterize them, and then we give the purified phage cocktail to the clinician along with any data that we have so that they apply for this expanded use through the FDA so they can treat patients. And when they are approved by the FDA, then they treat the patient. So because we are an academic institution, our phage program is guided by the basic science. So what I described to you earlier is how we could tailor phages for the gut that are specific for the gut, but we can also tailor phages that are specific for the drugs that these patients that are going to be on. And so this is work that Carmen has done in our lab, and what she has found using this assay she termed cynography is that certain phages and certain antibiotics work really well together. They can actually synergize. And it may be dependent on the bacteria, dependent on the phage, dependent on the actual antibiotic. So knowing this, when we develop cocktails for patients, we test our phage cocktails with the antibiotic that the patient will be on to see if it'll synergize so we can better tell the clinician what the best course of action will be. We also take a look at the resistance profile of some of these bacteria. So this is work that Keiko has done where she has characterized the X-Pac resistome. So what she has found is that these X-Pac E. coli have different mechanisms of resistance to some of our phages in our library. And she has characterized these genetically and phenotypically. But not only that, she's evolved phages that can actually overcome these resistant mechanisms, this HP3 to HP3.1. So knowing that, we can actually build cocktails that already have this phages that can overcome the resistant bacteria that we predict may happen. And beyond that, in the story that I just told you, what is happening at these mucosal sites, finding phages that are actually effective in these environments, the story that Jacob told you of these phages that are effective in urine. Well, that's also something to consider when we're building these phage cocktails. And so what work are we getting back, right? So this is work by Austin Terwilliger, and he has shown that in some of the cases that we're helping, particularly this UTI case, we actually utilize the knowledge that we gained from our previous studies. So this particular patient received HB3.1, the phage that could evolve to kill the resistant bacteria, ES17, the phage that was effective at mucosal sites, and ES19, a story that I did not talk to you about that is effective in biofilms. We utilized this cocktail, we gave it, the clinician gave it to the patient, and it was effective at eradicating a chronic UTI case. So this is how our basic science program can make a phage therapy program better. And this is the idea behind TAILFR, is at the center of it is, of course, helping the patients, but then also how our science is kind of helping build these cocktails that could be more effective, and the knowledge that we're gaining from treating these patients and can help our basic science program. And in other stuff I did not talk to you about is our technology too, where we're actually speeding up the adaption of these phages to better help these patients, as well as the purification processes of these phages so that these patients can get these pure, sterile, safe phages for clinical use. And then another leg of our program is, of course, outreach, is reaching out through social media, reaching out to clinicians, pharmacists, and regulatory agencies so that the patients, again, can get the help that they need. So I just wanted to acknowledge everybody in our lab and our phage team and our outside partners, and of course, all of our funding. And I will take any questions.
Thank you, Sabrina. If anybody has questions, they can type them in the chat or just speak up.
Yeah, I'll ask a question. Great talk, by the way. Thank you for that. So I wanted to ask, so with biofilms being a large problem in exacerbating the some of the issues that you talked about, how well do these phages work in bacterial strains that might perhaps be synthesizing some of these biofilms?
Yeah, so I don't do the biofilm work in our lab. A lot of this work is actually done by Dr. Belkey Sanchez in our lab, and she has been developing cocktails that are actually highly effective in these biofilm assays that we perform. So she performs MTT assays. So she's been characterizing cocktails that are actually really effective in these assays and testing them in animal model systems, similar to the animal model systems that Jacob talked about, these catheter UTI systems. And so that cocktail that I talked to you about that the patient received against the UTI was actually made up of the phages that Belkey's found to be very effective in these in vitro assays. And so that tells us that this is probably a pretty good assay, first of all, but also that, you know, what we're doing in the lab is actually what we're seeing in a patient too. So yes, we are looking into not only these phages' ability to kill the bacteria in these model systems, as well as in biofilms, as well, you know, as well as in at these sites too. I think I answered that question. I don't know.
I have a question. So do most of the phages that target X-Pac use capsule components as the receptor?
No, so I think Jacob talked about this. So they use LPS too, but yeah, they don't all use capsule as a receptor.
Mm-hmm.
We're pretty sure that ES17 is utilizing capsule as a receptor, but yeah, it can vary.
And are the phages that don't utilize capsule as a receptor more effective in the, like, your cecal fluid model?
So I've only tested, I haven't actually looked at what the receptors are for all of the phages that were in our library. So we have several phages in our library that we haven't fully characterized to see like what their receptor are. So I can't really tell you if maybe some of those phages were using capsule, but for some reason were not effective in mucin. They could be using a different capsule type. I don't know, that isn't similar to these mucin sugars. So yeah, I can't really answer that question.
Thank you.
Yeah.
Any other questions? Well, thank you so much, Sabrina and Jacob, and thanks to all of our attendees for coming and hearing about the cool science going on at Texas Med Center, and we'll see you next month.